Right, thank you very much for inviting me to come and talk. And what I wanted to do, because I just started here at the University of Manchester at the beginning of May, right, prior to that I've been at the University of Leeds for a long time. So what I wanted to do is just sort of give a bit of an overview of the work that my lab does around researching into defeating Alzheimer's disease. And I think about 18 months or so ago, we sort of came up with a mission statement for the lab to try and just sort of summarise in a nutshell what it is we're doing. And it's really is to understand the basic biological processes underlying Alzheimer's disease. That's what we are. We're cell biologists, biochemists, and so on in that respect. But then taking that on so as to identify opportunities for intervention and ultimately translate that research into practice that improves quality of life. Now you could probably almost take the bottom end of that and stick your topic area at the top of it, because ultimately one would argue that's what we should be trying to do in the work research we do across any spectrum. I realised that I haven't put in my slide which gives the numbers of individuals with Alzheimer's disease, the cost to society and so on. The figures that we've heard, you can start coming up with 820,000 people in the UK currently affected with dementia, of which Alzheimer's disease is the commonest form. The figures I've got, which is where the schizophrenia figures look interesting, the cost is £23 billion per annum. One of the things you can have problems with dementia is how you actually quantify the cost of care, because most of it is done within the community, within families and so on at that level. And what does it mean? Fundamentally, we are cell biologists. We're interested in the molecular cellular mechanisms underpinning the disease. So this just summarises what's known as the amyloid cascade, which is essentially saying this amyloid beta, a beta peptide, a short peptide, accumulates and then deposits in the brain. Post-mortem, you can see it, neuropathology is these amyloid plaques, but it causes lots of problems associated with nerve cell death, neurodegeneration, memory loss, etc. You can see there's a number of arrows on here, and there are question marks all over the place. Despite amyloid having been discovered back in the early 1980s, we still don't understand how all of this interrelates. Why does build-up of amyloid cause nerve cell death? Why do you see neurodegeneration in an individual when they're 70, 80 years old, when these processes may well have been happening since birth? So how does it all relate through? We know that in a minority of cases there is a genetic defect. It's only less than 5% of the cases of individuals with Alzheimer's disease. The majority of cases it's so-called sporadic, it's something else. Environmental, combination of minor genetic influences, head trauma, obesity, all these things could contribute to the instance of the disease. So we're interested in particular what regulates production of the amyloid peptide and how it then interacts and causes alterations in this other protein called tau, which you can then see in the brain in these neurofibrillary tangles. That amyloid beta peptide is a small peptide, only 40-42 amino acids, but it's derived from a larger precursor protein, the amyloid precursor protein APP. And it's cut out by two enzymes, the beta secretase and the gamma secretase, to release the peptide. That then aggregates and gets deposited ultimately in the plaques in the brain. That's a very, very simplistic description of it. There's also an alternative pathway over here, sort of anti or non-amyloidogenic, in which the precursor protein is cleaved by the alpha secretase, members of the adenines family, which is the reason why they're called that. It's got nothing to do with the TV programme. And that precludes the formation of amyloid beta. You'll be pleased to know that this pathway predominates in all of us all of the time, even under disease conditions. So we're interested in what's regulating these processes, and this is one of the studies we published a few years ago now, where we've shown that the cellular prion protein actually regulates this enzyme here and the production of amyloid beta from that point on. Prion protein is also known more famously, maybe, or infamously, as the causative agent of Creutzfeldt-Jakob disease, better known as mad cow disease. This is the normal cellular form, which all of us have throughout our bodies, particularly nerve cells. In mad cow disease, it undergoes a change in shape, and this is totally unrelated to that. So we're interested in what regulates these processing pathways. We've set up an siRNA, small interfering RNA screen, to identify novel regulators of this pathway. And we're also interested in actually activating this pathway. Because if we shift the whole thing more to the left, we will reduce the amount of amyloid beta, as well as cause a number of other protective effects. Activating things is not as easy as inhibiting them, that's the problem. Rob Andrew, PhD student, and Kate Kellogg, postdoc, have come across these with me, are both involved in aspects of this work. So I said that there are only just a few genes that actually have mutations in them that lead to Alzheimer's disease. So these are in the amyloid precursor protein itself, or in components of the gamma secretase enzyme. In the case of sporadic AD, there's the apolipoprotein E, which we know is a risk factor for the disease, but we still don't really know exactly why. And up until a few years ago, that was essentially all we knew about genetics of Alzheimer's disease. And then there's been several genome-wide association studies which have now resulted in some 22 new genes which are influencing the production, metabolism, action of the amyloid. And so genetics has really helped throw up, and this is just some of them listed there, a whole host of new genes which have implications in the disease process. The fundamental question is now exactly what those genes are doing, or their products are doing, at the molecular and cellular level. How do they influence amyloid beta production, etc.? And it's interesting questions where most of the time it's not actually mutations within these genes, it's polymorphisms. The other area we're interested in is when amyloid is formed, it aggregates together because it's a relatively hydrophobic, sticky protein. It then binds to the surface of nerve cells and causes problems inside them. And we're interested in identifying what actually it binds to at the cell surface and then how it communicates inside the cell. And basic questions around that. In order, if we understand those processes, can we then potentially intervene in some way and block that toxic signaling? Beth Noble and Hannah Griffiths and the other two members of our group have moved across with me, and they're interested in this area as well. And this just shows you can see anyway, sort of stuff we do. So this is fluorescence microscopy of rat hippocampal neurons. So you might just be able to make out in green the outline of the neuron, the blue blob is the nucleus in the middle. And basically what we're looking at here is the prion protein on the surface of the neurons. If we deplete those neurons of prion protein with small interfering RNA, you lose the staining. And then we've taken amyloid beta here, and you can see that only when we have this particular form of the amyloid does it bind to the neurons, and when we remove prion protein, we lose the binding. And that's quantitated up there. So prion protein also involved in binding amyloid. That raises a lot of questions, not least, well, how does that then signal across the membrane and cause the interaction with protein tau? What are the other proteins involved? And how does that signaling complex work? And we're interested in teasing that out at various levels. If we understand those sorts of processes, can we go in and intervene in them in some way? And this just shows you sort of proof of principle that maybe it's possible. So this is taking two compounds, EGCG and resveratrol. If you add them to those amyloid oligomers, you reduce the amount of oligomer that binds to the cell. And we again pick that up by immunofluorescence microscopy. It's quantitated there. Importantly, we completely lose the cytotoxic effects of the amyloid. So our EGCG resveratrol appears to be altering the shape of the oligomers so they no longer bind to the receptor protein, no longer cause the toxicity. You may have come across EGCG and resveratrol yourself. If you drink green tea or red wine, they are components of those. I'm not saying go away and drink loads of green tea and red wine, because you would not be able to drink enough to get high enough levels of these relatively minor ingredients in them into the brain to start having an effect. But there's no harm in having a bit of some of them. Personally, I prefer red wine.